On November 25th, 2018, it felt like the world changed. I just finished my dinner at my home in California, obsessively went to check my emails, and there was an email from a friend with the subject line, CRISPR babies. And I thought, ooh, this is going to be interesting. It actually reminded me almost immediately of an earlier Sunday, February 23rd, 1997, when as a relatively young academic, I got a call at home on Sunday morning from my dean. I wasn't accustomed to getting calls at home from my dean, let alone on Sunday morning. And I was wondering what I had done wrong when he said, Hank, they've cloned a sheep. I thought you should know. And then too, I thought things are going to get interesting. In both cases, I was right. And I want to talk about the November 2018 events and what they mean in terms of what we should be thinking about with respect to human genome editing, human DNA editing, the science of it, and the ethics of it. Specifically, I want to talk about three relatively non-controversial uses. Then I want to talk about the most controversial use, so-called human germline genome editing, and whether it's a good thing, whether it's a bad thing, whether it's going to be an important thing. And then finally, I want to talk about some of the broader lessons I think this has for the relationship between science and society, lessons that I think are quite important. So let me start by talking about CRISPR and some of its potential uses. I don't think I have to tell this audience much about clustered, regularly interspaced, short palindromic repeats, other than to point out that the inventors of CRISPR did not win the Nobel Prize this past year. Those inventors were anonymous bacteria 1, 2, 3 billion years ago. But Jennifer Doudna and Emmanuel Charpentier, I think, fully deserved the Nobel Prize for turning this bacterial defense mechanism into a fascinating and really versatile human tool. It's not the first way we've been able to edit genomes. Arguably, that goes back to Paul Berg and recombinant DNA. And in recent years, things like TALENs and zinc finger nucleases have done a fine job of editing. It's just that CRISPR was faster, easier, cheaper, more accurate. It's like the change from early automobiles to Model Ts. Early cars were terrible and they broke down all the time. They were expensive. You needed to be rich and have a chauffeur/mechanic. Model T, anybody could have it. And pretty soon anybody did. And that's what changed the world. CRISPR is going to change the world, not because it's a new thing that we can now do, but it's because something that we could do with great difficulty can now be done with much more ease. There are three uses for it that I think are going to be relatively non-controversial. The first is to use it to edit human cells for in vitro research, whether it's individual cells or more likely cell lines or even organoids, collections of 1 million or 2 million cells that are forming structures that seem to be trying to become bits of intestine or bits of heart or bits of brain. We do this in order to mutate a gene, knock out a gene, add a gene, and see how it affects the functions of those cells and the proteins those genes make. I don't think anybody is going to worry very much about the ethics of doing that. Second is a little bit more problematic, but not much, and that's what's called somatic cell gene editing. Somatic cells are almost all of our cells. I have approximately 35 trillion cells in me, or I should say 35 trillion human cells. I've got another roughly 35 trillion non-human cells. Of those 35 trillion cells, approximately 34.999 billion of them are somatic cells. They are my soma, they are my body. When I die, they die. Most of them will actually die before I die. What distinguishes them from the other cell type, the germline, is those cells arguably don't die, or at least they may not die. Those are the cells that in me are sperm, or are cells that then make sperm. In women, they're eggs or cells in fetuses and embryos that ultimately become eggs. That's the germline, and it's different from the soma. And that changes to the germline can get passed on to the next generation. Changes to the somatic cells cannot. Now, of course, They can affect the next generation. Keeping somebody alive who wouldn't have lived has major effects potentially on later generations, but they're not genetic sequence effects. So somatic cell therapy, somatic cell editing, also known as gene therapy, is less controversial. It's been tried for over 40 years. It's finally paying off. Both European and East Asian and American regulators have started approving gene therapies. The 40 years of development actually didn't have that much to do with editing techniques. There were questions of delivery and of the pathogenicity of the delivery mechanisms. There were all sorts of difficult details that needed to be hammered out, most of which got hammered out by the time CRISPR came along as a tool. So gene therapy is exploding right now, but it's not particularly exploding because of CRISPR. I think CRISPR is going to be used for it for the most part because it is cheaper, easier, and faster. But a lot of the work was originally done with TALENs or with zinc finger nucleases. CRISPR for gene therapy through somatic cell editing is not controversial. It does raise ethical questions, but they're not different ethical questions than those we confront with any new technology, biomedical technology, particularly an expensive one. There are questions of when is it ready to try? Who should we try it on first? How expensive is it going to be? Who's going to pay for it? Who's going to have access to it? If there's a limited supply, who gets it first? If there are people who need it who aren't able to consent because they're babies or they're embryos or fetuses, or they're people with mental problems who can't consent, how are we going to handle that? Those are important questions, and they're going to be questions for gene therapy. Certainly the price tag question is a huge question for gene therapy because they're getting huge price tags, but they're not novel questions. They're questions that we encounter all the time in biomedicine. And, do we get them right all the time? No, but we do a decent job in handling them. So in any event, although there are ethical questions, there are not huge ethical controversies about these. Now, the third category is also not controversial, not very controversial, although frankly, I think it should be a lot more controversial. And that's using CRISPR or other genome editing on non-humans. I think this is really the most important thing to watch because we're going to do it much more quickly and easily. If you want to try to edit people and in 5% of the time it leads to death, deformity, disease, disability, things like that, that's not good. That's not acceptable. If you do it in dogs and cats and 5% of the time it leads to bad kittens or puppies, it's unfortunate. We'll feel bad. If it's cattle or pigs, well, we'll feel a little less bad. If it's mosquitoes, we won't feel bad at all. The thing we'll feel bad about is it didn't kill off all of them. So we're going to be much more willing to try this with non-humans. The regulatory schemes are complex and different from place to place, but often simpler than the schemes for medical uses. And so I think this is something that we do need to pay a lot of attention to, particularly since the regulatory schemes we have, I think, are grossly inadequate. We will be able to edit the entire biosphere, and we're likely not to start with humans, but start with others. However, because we're humans and a very egocentric species indeed, we're paying most of our attention to the humans and not watching the other 99.9999% of life forms on the planet. Which leads me to the second major point I want to talk about, and that's the issue that we are paying attention to, and frankly, I think probably too much attention to. That's human germline genome editing. Let me start by explaining what the heck that means. The human germline is eggs and sperm, and the cells that give rise to eggs and sperm, and because they're eggs and sperm, they can pass on to the next generation. Now, editing that potentially edits the next generation and every generation to come. It is important, though, to recognize that that's only potentially. It is entirely possible that someone won't have genetic children. It happens a lot. Or that their genetic children won't have genetic children, or that their genetic children won't have genetic children. If I look back to the siblings of my great-grandparents, The majority of them do not have living descendants at this point. Either they never had children, or their children didn't have children, or their grandchildren didn't have children. So most of the time, it's probably going to be a dead end, but not always. And in any one case, it might go on for generations and generations and generations. And that's what's prompted the great controversy about germline editing. It's not just done and over in one lifetime. It could potentially expand a lot longer. There is history to this. Back in the early 70s, when recombinant DNA came along, and the Asilomar conference was held, many of the scientists there said, well, don't worry about this recombinant DNA. We'll never do the most striking things. We'll never do germline editing. Germline editing had been talked about fairly seriously since the 60s, and not so seriously in science fiction since before then. And some scientists promised, no, we're not going to do this. Now, it wasn't science that promised it, and it wasn't a binding promise, but it was in the air. Unfortunately, promises made to not do something that you don't know how to do and don't know whether you'll ever know how to do are not necessarily always made with the greatest of forethought. Easy to promise not to do something that you can't do. I promise not to fly out the window and fly from here to Baltimore, flapping my wings. That's an easy promise for me to make. But if I started getting wings and I could do it and I really wanted to go to Baltimore, then the promise becomes maybe more binding and more significant. And CRISPR made it more present. CRISPR didn't itself make possible what was previously impossible, but it made much more plausible and much more likely what was previously implausible, unlikely, very difficult. To her great credit, I think, Jennifer Doudna called together a meeting of about 14 people in January 2015 in Napa Valley. I was lucky enough to be one of those. We reached a consensus fairly quickly, which ended up getting published in Science, that basically said, don't do this now. This is way too risky to try to edit human embryos, to edit the germline. And let me note, you could edit the germline in a number of ways, including editing eggs and sperm. But what I'm talking about, people have talked about, what's mostly controversial is editing embryos, IVF embryos. And we said, it's just insanely risky to the embryos. And besides, we should talk. We as society and as scientists should figure out, A, whether it's safe, B, whether it's effective, and C, whether it's useful, what we should use it for, what we shouldn't use it for, what society wants us to do with it. And that was pretty much a consensus. A number of national and international bodies came out with recommendations that went up and down, but basically said the same thing. Don't do it, at least don't do it now. And before you even think about doing it, make sure it's safe and effective. And then you've got social consensus, social permission, social acquiescence, social engagement, social discussion, that society is okay with you doing it. That's pretty much where matters stood on November 25th, 2018, when we all learned in a story broken by a very enterprising reporter for MIT Tech Review, Antonio Regalado, that a Chinese scientist whose name I'm going to butcher, but is something similar to He Zhonghui, was discovered to have edited human embryos and brought 2 of those embryos to term in the form of 2 non-identical twin baby girls. He let the story out after Regalado had broken it on that Sunday night, Monday morning in China. 2 days later, he presented in Hong Kong at the second International Summit on Human Genome Editing. He talked for about a half hour. He presented his findings. He took some questions and answers. He left the stage and has not been seen in public since then. He went back to China. He was apparently placed under house arrest. We think we know from a Chinese government press release that in December of 2019, a little more than a year later, he was convicted of various crimes and sentenced to 3 years in prison. As far as we know, no outsiders have seen him since he left that Hong Kong meeting. We also don't really know what he did. We know what he said he did in Hong Kong, some of which appears to have been false. He said the babies were born in November and were healthy. It really looks like they were born in early October, quite premature, and spent quite a while in the neonatal intensive care unit. We don't know exactly what changes he made or what changes he was trying to make. And there's a third baby. The only thing we know about the third baby is in the press release, in the Chinese government release about his sentencing, it said a third baby was born in the summer. We don't know what it was edited for. We don't know whether it's male or female. We don't know its health. We don't know anything. And that's one of the frustrating things about this story. There's so much that we don't know, and the Chinese government does not seem to be in any hurry to help us learn. Now, I don't want to spend much more time on He Zhonghui. I published a book this February with MIT Press called CRISPR People, The Science and Ethics of Editing Humans. And about half of the book is devoted to the He Zhonghui story. I will say, I think it is criminally reckless. It was a criminally reckless experiment. My only complaint about his 3-year prison sentence is I wish it had been longer, and I wish I knew what kind of prison he was in and what he was doing. He was breaking a broad international consensus. He was grossly risking these babies, these future babies' lives for almost no benefit and really incalculable risk. He was doing it while informing the parents very poorly about what was happening. He apparently was actually bribing and lying to Chinese government authorities, and he was keeping the entire thing very secret. This is some of the most unethical research I can imagine. So I'm not unhappy about him. It's a fascinating story, and I recommend my book, and a number of other books and articles are out there about this saga. But that's not what I want to talk about today. What I want to talk about is germline editing more broadly. Is it inherently a bad thing? Is it likely a good thing or not? And then what does it say about science's relation and what the relation between science and society is and what it should be? So let's start with human germline genome editing and whether it's inherently a bad thing. Most of the national commissions, the US National Academy of Sciences, the Nuffield Commission, the Nuffield Commission in the UK, the German ethics body, ethics committee, they all came out with things that said, don't do this now. Maybe you could do it if it's shown to be safe and effective, but you need good ideas about what to do and what not to do, and you need social acceptance, consensus, acquiescence, engagement, some sort of permission from your society before you go ahead with it. A commission set up by the US National Academy of Sciences and the Royal Society and others came out last year with similar kinds of recommendations. So that's saying, don't even think about it unless or until it's proven safe and effective. Research that I think should take at least a decade. But there's another view which says, even then, don't think about it. And this view is germline editing is a line that we should not cross. Now, why shouldn't we cross it? Well, because we're playing God, because we're playing Darwin, because we're playing evolution, because we're doing things that we're not wise enough to do, because we are meddling and doing things man shouldn't do. It's a version of Prometheus and, of course, Frankenstein, the subtitle to which was The Modern Prometheus. That there are, or a version of the tree of the knowledge of the forbidden tree of the knowledge of good and evil. It's an old story, and many people feel it very deeply. I don't find it very convincing. I don't find the story very convincing in general because our entire civilization has been built up by doing things that are unnatural, doing things like keeping people alive who would otherwise die, or having Zoom meetings, which nature probably didn't intend. But in this context, I think there's another reason to be really skeptical about it. I think it's based in part on this sacralization of something called the human germline genome. I don't think it exists, at least not the way people think it does. There's this sense that people have that the essence of our species, the ark of our covenant, is this sacred human germline genome that should not be messed with. but that's not reality. There is no the human germline genome. There's 7.5 billion of them. Mine is different from yours. Identical twins have slightly different genomes. Which one is the human germline genome? Well, obviously it's mine, but other people may disagree with that. Not only that, but it's changing every generation. I don't have exactly the same genetic sequences that my parents got from their parents because of mutations that happened between the time that their parents conceived them and the time they conceived me. My kids don't have exactly the same genetic variations that I got from my parents because of mutations that happened during my lifetime in my germ cells. The germline is changing. It's changing from generation to generation inherently. There's no way to stop it. It's also changing in a different sense, in a way that humans influence, if not control. And control may not be too strong a word for it. When we change the environment, we change, when it's relevant genetically, we change the prevalence of different genetic variations. So there are genes that help us digest starch. Modern humans have several copies, almost all have several copies of some of these genes. Our hunter-gatherer ancestors before farming had fewer copies. What's the story one can tell? Well, the ones who had extra copies as a result of some duplication event digested starch better. They were healthier. The kids that they passed those genes on to were healthier, and pretty soon it got fixed into society. We see this in smaller ways in things like adult lactose tolerance, which in some populations, and not just in Northern Europeans, it appears to have been independently evolved 7 or 8 times in humanity. People end up being able to digest milk and milk products in their adulthood without gastric distress, something very unusual in mammals. And so at least in some societies, that's become really common. I have lactose tolerant genes because the environment my ancestors grew up in favored people who could drink milk because they had big dairy industries, drink milk, eat cheese, and so on. Or maybe most powerfully is medicine. When medicine stops a genetic disease, it's changing the future prevalence of different alleles. Until about 100 years ago, and a couple of, interfering playing God Canadians named Banting and Best realized that you could inject insulin to keep type 1 diabetics alive. They all died. They died quickly, and they almost all died before they could reproduce. It was called juvenile diabetes for a reason. Now, juvenile diabetes is not powerfully genetic, but there are HLA haplotypes that predispose one pretty strongly to it. Compared to the rest of the population, a couple of orders of magnitude of increased risk. Those versions are almost certainly more common in the population now because people who carry them, who got type 1 diabetes, can now reproduce. My cousin Mike, who's exactly my age, got type 1 diabetes when he was 9. He now has either 13 or 14 grandchildren. He's not quite sure how many, but he wouldn't have had those grandchildren but for Banting and Best and modern medicine. Medicine changes the genome. So I just don't see anything really special about the genome. Yes, it has potential effects in future generations, but everything we do has potential effects in future generations. Our technologies have effects in future generations. Certainly the increase of CO2 in the atmosphere, we're screwing over future generations that way. We're affecting future generations all the time. I don't see this, particularly when it's mainly being used to avoid disease, as being particularly troublesome. Now, here's where many people have a strong visceral reaction. They're worried about it, not for disease, but for enhancement, for super babies or the X-Men, and especially for rich people having super babies who become a genetic nobility. It's a good storyline. It's been used a lot in science fiction. I like science fiction, but I don't make policy based on science fiction. And I think this is science fiction. Enhancement is not something we know how to do. We know of hundreds of genes that powerfully affect intelligence. If the gene is broken, the person has very low intelligence. We know of almost no genetic variations, maybe none at all, that enhance intelligence. Even the most optimistic people in this field currently see maybe 1 or 2 IQ point difference, and others disagree with that. We know we can make that difference by better childhood nutrition or reading to kids or better childhood health. You know, the super athlete, the super musician, the super math genius, the super intelligent person, 30, 40, 50 years, maybe we'll get good enough at understanding the 23,000 genes and all the ways they overlap with environmental influence in different variations, and all the ways chance, particularly in brain development, affects them. I'm a little skeptical myself. We'll certainly know more than we do now, but will we know enough to be able to, use genetic engineering to add 30 IQ points to embryos? I'm deeply skeptical. But in a way, it doesn't matter what I am. It's not going to happen in my lifetime. I'm perfectly happy to kick the can down the road 50 years, 100 years, 150 years, when the people who are alive then will do whatever it is that they think should be done. And they're not going to care what we think should be done. To eliminate an entire field because 50 or 100 years from now, people might make decisions we don't like seems to me a bad idea. So I am not in the camp that thinks genome editing is inherently bad. On the other hand, I don't see hardly any good uses for it. Almost everything you would want to accomplish, except enhancement involving multiple genes, can be done in other ways. And enhancement we can't do anyway. We can do it in other ways for single genes through pre-implantation genetic diagnosis. Instead of editing out bad genes in embryos, we select embryos that don't have those bad genes. That's been clinically available, safe and effective for over 30 years. Or increasingly, we could let the babies be born and use somatic cell gene therapy to fix the problems. When would you need this? Very, very rarely. If you've got a couple where they both have an autosomal recessive disease, they manage to live long enough and be healthy enough that they want to have kids that are genetically theirs, but they don't want them to have their disease, well, they'd need to use this. Is there any good reason to stop them? I don't see it, assuming it's safe and effective. Is there any reason to think there are going to be a lot of them? No, a handful. So I don't see a big use case for this unless or until enhancement becomes plausible. And I think that's probably after the lifetime of almost everybody listening to me, if ever. So, I seem to have this tendency in my work to pick a position that makes enemies on both sides. So I get shot at from the people who think it's inherently bad, and I get shot at from the people who are excited and think we need to start using it. It means I don't have to worry which way to look for the incoming arrows because they're coming from everywhere. But that's my best look at it, my best understanding of what it's likely to be. Not a big deal either on the negative side or on the positive side. But it is a big deal in some other respects, and that's what I want to end on. I want to end on a call for greater scientific humility. This has exposed rifts between society, which is very concerned about this, and at least some people in science and bioethics who are concerned about safety and efficacy, but are less concerned about it going forward if it's safe and effective. Now, I think safety and efficacy are immensely important ethical concerns. It is unethical to do unreasonably unsafe or ineffective things to people. But there are other concerns as well. And if society wants to ban something, that's its right. Science doesn't get to make the rules. Science presents opportunities and alternatives. It explains them. It tries to predict various consequences. But ultimately, if the people, either in their individual contexts as parents, prospective parents, or collectively as governments decide they don't want it, science needs to agree. It has to. I think in part it should through political theory. I think the people rule, Plato's philosopher kings never happened, and science kings and queens are not likely to happen and shouldn't happen. We should have government of, by, and for the people. But even if you didn't believe that, there's no choice. Science cannot exist without the support of its society. It can't exist without grants. It can't exist without jobs. It can't exist without social status that attracts people to it. Rogue scientists are much more common in science fiction and in myth than they are in reality, because most scientists need grants. They need approval. They need a job. They need things that going rogue is likely to deprive them of. He Jiankui actually was in a very interesting situation of having kind of free and unfettered money that allowed him to do what he did. That's very rare. So science needs to understand that it is a servant of its society, and it's a servant necessarily because it can't help but be. It needs society. If South Dakota says it's a felony to do human embryonic stem cell research there, science can say, you know, we think that's a bad idea. Here are a bunch of reasons we think you're making a mistake. But if that's what they say, as they did, you move to California, which will actually fund your research. So I think science needs to be more humble in its approach to society. And we've seen some of this in the aftermath of the He Jiankui affair, whose story evokes the myth of Frankenstein and Prometheus. It leads to concerns about rogue science, and the fact that a number of researchers, scientists, academics around the world knew about this and didn't tell anybody doesn't make the story any better. The fact that it came as a great shock to people, there was no discussion in advance, increases the damage that's done to science by it. I care about science. I think science has the potential to do wonderfully good things, many more good things than bad things, but it can only do that within and as part of its society, not in opposition to it. I think the He Jiankui affair provides another example of that. Thank you very much for your time and attention.